This has really been a great trip, and I've enjoyed also the series of previous talks, which have all sort of introduced what I'm going to talk about today, which is gene-specific DNA methylation in Down syndrome blood and in Down syndrome brain. And I'll also touch on diet, as Rima and Jill already have. So Jill certainly introduced what epigenetics is, and also the concept that the two main epigenetic marks on the DNA are histone modifications, about which we're not saying much today, and DNA methylation. So why study DNA methylation in particular? It is a fundamental epigenetic mark. When promoter regions of genes are methylated, it's a very effective way to silence the gene. When other regions in the genome get methylated, like insulator elements, for example, this can actually activate gene expression. So one does not expect to see a one-to-one correlation between gain of methylation and loss of gene expression. It depends where the methylation occurs, that is, the precise pattern of methylation that has changed. It is a phenomenon that, as you already have a feeling for, is perturbed in diverse human diseases, although a lot of the research on DNA methylation started with funding from the National Cancer Institute in the US. And now one of the NIH roadmap items is epigenetics in non-cancerous diseases, including Alzheimer's disease and autoimmune disease as well. And importantly, again, Rima and Jill have shown that methylation can be manipulated by nutrients, and of course we also have drugs that can change methylation. The genetic-epigenetic interactions that I'm going to talk about are certainly central to this symposium. That is, we know the cause of Down syndrome, it's trisomy 21. It's a genetic cause, but we don't know the pathogenesis of the complex phenotype of Down syndrome. And I would argue that part of that pathogenesis is an epigenetic consequence of the trisomy. So DNA methylation is transmitted to daughter cells. This is something that Jill didn't cover quite as much, but this DNMT1 DNA methyltransferase enzyme actually has a very high affinity for hemimethylated sites. So when the DNA replicates, you end up with a hemimethylated site, you can see here, and the enzyme goes right there. In fact, it is already in the right place at the right time to remethylate that site and propagate the pattern. But these patterns can change with environmental manipulations, and one of them that's used in cancer treatment, particularly in leukemia, is this drug 5-AZA-DC, DAC, or decitabine, which is in the US FDA approved for use in myelodysplastic syndrome and is also used in AML as an anti-cancer agent. And for chemoprevention of cancer, people throughout the world now are becoming increasingly interested in folic acid, B12, betaine sort of diets for chemoprevention. And I was actually personally skeptical of the ability of diet to change DNA methylation in a biologically important way until in my own lab I was convinced by one of the fellows to do the experiment and pay for the expensive mouse diet to feed mice. And this is a mouse stomach cancer model, very similar to human stomach cancer in its pathogenesis. These are it's called in mouse Helicobacter felis, but it's analogous to Helicobacter pylori in humans, and these are hypergastronemic mice. So it's a very faithful model for gastric cancer. And when he started feeding the expensive diet to the mice at weaning, very early, right away, the gastric dysplasia was completely prevented, and you see that in the stomach in part D there. Whereas if he didn't supplement the diet and the mice were simply on a normal diet, without the extra methyl donors, you had this severe dysplasia and microinvasive cancer, which you see in part A. So it was actually a very potent chemopreventive regimen, which prevented the loss of methylation that one sees with gastric cancer. And this is just a bar graph showing that. And prevented the inflammation. So gastric cancer is an inflammatory mediated type of cancer. It is associated with H. pylori infection. And indeed, the number of CD3 positive T lymphocytes in the gastric mucosa was reduced by the methyl donor-rich expensive diet. So that's what, just to say that I very much agree with what Jill was putting forward, that diet matters. And I do think that in some circumstances where you do have Burger King and McDonald's, it might not be enough just to make dietary recommendations, but one might want to think about supplementation. But I basically agree with what Jill was saying. So what about DNA methylation in Down syndrome? I'm not going to cover the interactions in cis here. We have a trans interaction between a genetic change, trisomy 21, and an epigenetic modification of the DNA methylation. In the field of epigenetics, in contrast to pure genetics, it is important to clearly specify what cell type one is working with, because we have in our body a single genome, but probably as many epigenomes as we have cell types. And so one cannot assume that everything that one does on a blood sample is directly relevant to the brain when one is working on epigenetics. One can assume, though, that everything that one does using a blood sample and analyzing epigenetics is relevant to the blood. And in fact, there are well-documented and important blood-related phenotypes in Down syndrome. There's altered lymphocyte and natural killer cell function, manifesting as a very strong increase in certain types of autoimmune diseases, including celiac disease of the intestine, alopecia areata, a type of autoimmune hair loss, and thyroid disease probably as well, accounting for the hypothyroidism in some cases. And there are recurrent infections in Down syndrome, both viral and bacterial, above the frequency of the non-Downs population. So we started looking at methylation, profiling it on microarrays using blood samples as our first step. And we found a limited set of genes, on the order of 100 genes, with different methylation in Down syndrome in case after case. This is a very reproducible finding. We have 30,000 genes. Only about 100 of those are altered in their methylation in the promoter region in Down syndrome. But those same genes are affected in case after case. And that's essentially what these heat maps show. These are heat maps of DNA methylation in promoter regions in Down syndrome blood versus control blood, which are age matched. In AML, I'm just showing a few cases of leukemia, but for contrast here, to show that the genes affected in leukemia are a completely different pattern, and they're affected in a more drastic way. There's a darker red, a darker blue color. The changes in Down syndrome, these are blood samples not from Down syndrome with leukemia. These are from actually adults where there is no issue of leukemia. In fact, adults with Down syndrome are resistant to cancer by and large. But these changes are nonetheless, they're milder but very reproducible. Okay. When you find a gene with altered DNA methylation in Down syndrome, it can be very dramatic. There are not very many genes affected, on the order of 100 genes, but this is a very dramatic example. In case after case of Down syndrome, this gene in this internal promoter region is hypomethylated. The white circles are CpGs that are unmethylated, the black circles CpGs that are methylated. It's a very dramatic finding. You can see it by a gel-based method on the top. We can quantitate the methylation using methylation-sensitive pyrosequencing, and here the best marker is this one here. Here are the non-Downs normal blood samples. Here are the Down syndrome blood samples. This is an interesting gene because with age, in the normal samples, you see this decline in methylation, and the Down syndrome samples appear, in this sense, prematurely aged. They all have the hypomethylation. But other genes, which are also highly statistically significantly different between Down syndrome and normal samples, do not have this age effect. So if you look at this gene, which is CD3 zeta, This is the major encoding the major signal transducing subunit in T cells. There's a very statistically strong difference between normals and Down syndrome. And for many, though not all, of these genes, the methylation affects expression. We can see a difference in expression of the gene in DS versus control for these two loci. And in fact, we can manipulate expression of those genes. I'm sorry. In fact, we can manipulate expression of those genes in cell culture with the demethylating drug 5-AZA-DC, and they move in the expected direction. Okay. So now we've become even more precise in the cell type that we're working with, because blood, although it's a defined tissue, is nonetheless heterogeneous in terms of its component cells. And we've started to purify CD3 positive T cells from our blood samples, and we've accumulated quite a nice series of these highly purified effector T cells. And again, we see about 100 genes differentially methylated, some with relative losses of methylation and other genes with relative gains of methylation in Down syndrome. If you tell me the name of a gene, I can tell you this gene is, in case after case, shows loss of methylation. If you tell me another name, perhaps this gene, in case after case, shows gain of methylation in Down syndrome. But it's a limited set and very reproducible. Here's a cute method where we do copy number analysis using the same microarrays, and you can see loss of the intervening sequence in the T cell receptor. So we know that most of our preps are quite good in terms of the T cell purification. And again, very dramatic. This is an interesting gene, ZDHHC14, which encodes a palmitoyltransferase, which is essential for targeting the Abl kinase to the cell membrane. So this gene, no doubt, plays an important role in T cell signaling. And the loss of methylation here in the promoter region is quite impressive, where you have almost no methylation in Down syndrome and strongly methylated on most of the alleles in controlled T cells. Here's an example of manipulating the expression of that gene. So if I go back, here you have the controlled T cells, very densely methylated, and there's a loss in Down syndrome T cells. We can take control T cells in culture. These are not leukemia. These are normal T cells. We can make them divide with PHA, add the demethylating drug 5-AZA-DC, and you see that this is a methylation-dependent expression because we can reactivate expression of the gene with demethylation. Okay. In the normal cells. So schematically, we have a situation where you have trisomy 21, a genetic change. There are altered regulatory networks downstream of that genetic change. Cellular selection, because of course the blood is a stem cell dependent organ. So we have turnover of these cells all the time in the bone marrow. And ending up with an altered pattern of DNA methylation in Down syndrome, altered in a stereotypical way in person after person. If you have trisomy 21, you're likely, very likely, in fact guaranteed, to have loss of methylation in the TMEM131 gene and loss of methylation in ZDHHC14 gene. So there's outgrowth of these clones, and the question is, for this symposium, with a change in diet or with methyl donor supplementation, could some of these problems be corrected? And I think this is a very doable study. We call a study like this a clinical trial. You have a support for this, preclinical support for this idea, and it would be an interesting and benign, at the very least, type of clinical trial. Because, as you know, certainly as the parents in the room know, many children and young adults with Down syndrome are already getting vitamin supplements from mom and dad. What we're proposing here is perhaps a controlled study of that type. Okay. We know what to look for in the lab because we have very well-defined epigenetic abnormalities. Measurable. What about the brain? This is what I just said. So, of course, cognitive disability, early onset Alzheimer's are important problems. Through an amazingly productive collaboration with Jurgi Wengel at the Institute for Basic Research in Staten Island, to obtain a series of autopsy brain samples from Down syndrome adults is like gold. But he has, over the years, banked these brains. And Gudrun Moore, who we'll hear from next, has created a similarly valuable brain bank from fetal cases. So we've been able to take this material, either macro dissect the gray matter and make DNA, or as I'll show you, even purify neurons away from glia and from this archival material and profile DNA methylation. And again, we see very recurrent stereotypical changes in a limited set of about 100 genes, albeit a different 100 genes from the genes that are affected in the blood. And the striking finding in the brain is that almost all of the changes are gains of methylation in Down syndrome, whereas in the blood, a number of very important, physiologically important genes, as I've shown you, have losses of methylation. Here's a denser array showing exactly the same phenomenon. Almost all of the changes in brain cells are gains of methylation, red in Down syndrome, blue in the control. This shows our procedure for purifying neurons away from glial cells from the archival brain material. And it really works quite well. So we make a nuclear prep from this material and then stain the cell nuclei with a monoclonal antibody for the NEWN antigen, which only labels neurons and not glia, and then we flow sort the nuclei. And our final preps, here you see the NEWN positive. These are all neurons. They're all green. Superposition with the DNA itself gives this yellow color. For the NEWN negative prep, there are very few neurons left after we do the cell sorting. These other cells are predominantly glia. And here I'm showing you a validation of the methylation results from the array using bisulfite sequencing in a gene that happens to be on chromosome 21, although most of the affected genes are not. And we see the abnormality in both neurons and glia. So here you have a control brain, unfractionated DNA from the whole gray matter, and then the neurons and the glia. And this region of the C21orf56 gene is unmethylated in control brain cells. In the Down syndromes, I showed you that almost all the epigenetic changes in promoter regions in Down syndrome brains are gains of methylation. And we see this gain in these two individuals, and it's present both in the neurons and the glia, implying that this epigenetic abnormality probably occurred very early in brain development before these two cell lineages diverged. It's a very early event. So, the methylation abnormalities in Down syndrome are highly tissue specific. Altered methylation across the genome is consistent across individuals within a given tissue. These are all different individuals. They show a similar pattern relative to the controls, but different genes are affected in different tissues. And this raises the question, I think it's worth reading this slide, which is actually my last slide. Will methyl donor supplements be beneficial for brain development? It's a very interesting question because pregnant women sometimes take methyl donor supplements, notably folate. If the gains of promoter methylation in Down syndrome brain cells are a good thing, if these changes are acting to normalize gene expression, then the answer is yes. But if the gains of promoter methylation in Down syndrome brain cells are a bad thing, acting to perturb normal gene expression, then folate supplementation early in brain development in Down syndrome may not be a good thing. And this is a very interesting, to me, this is a very interesting basic research question. I want to be clear about this. This is just a partial list of our collaborators. We're also collaborating, as you'll hear, collaborating with Gudrun Moore, who's going to show actual data from that work. But to go back to this this question, I would just throw out, and this may be not correct, but I think that all the pieces are in place to do a controlled study now of methyl donor supplementation in young adults and older kids with Down syndrome with an endpoint of measuring improved immune function. And possibly secondary to that, there might be improved learning or cognition. I don't think we're ready yet, simply based on these data, to think about changing our patterns of folate supplementation in a woman pregnant with a fetus with Down syndrome or very early in the neonatal period in Down syndrome. And again, this is because we see this gain of methylation in the promoter regions in the brain. Overwhelmingly, these are gains of methylation. And this is such a critical developmental process that I'm not sure we're ready to do a clinical trial early in life. But later in life, again, with a defined period of several months of supplementation, for example, and very well-defined endpoint of normalizing the biochemistry and the methylation in the blood cells and measuring improvements in immune function, that seems to me to be a very feasible study. And so it's perhaps some of you will disagree with that, but I'll end with that idea.